It's a great pleasure and honor for me to introduce today's Grand Round speaker, Dr. Jin Chen. Um, Dr. Chen received her medical education at Shanghai Medical University and specialized in pharmacology at that time. She then proceeded to go to Boston in 1986 and received her PhD degree in biochemistry and molecular pharmacology from Harvard University. She did some postdoctoral work at MIT and then at Vanderbilt University. And at the end of her postdoctoral appointment, she was offered a faculty position at Vanderbilt University, where she is currently a professor of medicine and cell and developmental biology, and also is director of graduate studies in cancer biology. She is also a senior research career scientist in the Veterans Affairs at Vanderbilt. So she has a dual appointment, just like I have. Now, Dr. Chen has pioneered studies of the afferent receptors, it's called FA and FB, and this is a large body of receptors with receptor tyrosine kinases, and she has pioneered studies of the function of these receptors in cancer biology, metabolism, and angiogenesis, tumor angiogenesis. She was, in fact, one of the first to show that FA2 is involved in the angiogenesis in tumor cells. So she has a very productive and intensive research program at Vanderbilt. And so it's my pleasure to introduce her today to present her talk on this subject area. The title of her talk is Enhancing Anti-Tumor Immunity by Regulating Vascular Signaling and Selective Targeting of Tumor Glutamine Metabolism. Dr. Chen?
Thank you very much for the introduction and for the invitation and opportunity to speak here in the department seminar series. I actually visited the University of Minnesota actually a long time ago when I'm looking for assistant professor position. That was, I think, more than 20 years ago. So hopefully when the pandemic is over, and I will visit again in person. I was very impressed then. I'm still impressed now by the university and the research environment. So before I start, try to see how do I advance. Yeah, so before I start, I'd just like to introduce you the Vanderbilt University Medical Center. We are located in the Nashville, Tennessee, the Music City. We are very green, as you can see. Right now, the flowers blooming in spring. So any of you are tired of cold, please move down to Nashville. This slide showing that the lab gathering before the pandemic, we are having learning how to making sushi in a restaurant together with the Boothby Lab, which is our collaborator. We had a great time, and hopefully we can do this again very soon when everyone is vaccinated. So during the past 20 years or so, my laboratory has very intense interest in investigating the tumor microenvironment. As Khalil in introduction said, that we are initially actually focusing on the tumor cells and tumor blood vessel interaction, mainly studying FA2 receptor tyrosine kinase. And more recently, we go down the signaling pathway and focusing on mTOR complex 1 and complex 2, not only in the tumor cells, tumor blood vessel, but also we try to study the impact of those interactions on the immune microenvironment. So during the course of these studies, we discovered actually FA2 is regulating glutamine metabolism in the tumor cells, So that's why we also get into the metabolism field. But for today's talk, today's talk, I'm going to present two stories, which recently just published. The first one is spearheaded by Xiang Wang, who is a former postdoc in my lab, and she investigated the tumor blood vessel mTOR complex 1 activity and how that affects anti-tumor immunity. And the second story is based on the concept that the tumor cells and lymphocytes using very similar, or I should say the same nutrients for their proliferative program. So there is competition, and we study how glutamine metabolism in tumor cells and affect the immune environment. Okay, for the first story, I guess you guys already know that tumor blood vessels are really abnormal. On the left side are the scanning EM picture showing normal and tumor vessels. As you can see, that tumor vessels have lots of angiogenic sprouts, but they are chaotic. So the perfusion actually is poor. If you look at the inside the vessel in the lower panel, as you can see, there is a lot of holes in the tumor blood vessels, so causing the blood leakage, which increase the interstitial pressures, which impeding infiltrating lymphocytes. And also because of this choatic angiogenesis, and basically the perfusion is not good, so there is hypoxia within the tumor. Hypoxia increase the tumor cell motility and increase the cancer metastasis. So on the right is the immunofluorescence and showing the green, which is CD31 staining. So there is a very green tube, and on the normal situation, it's tightly wrapped around by parasites, which staining red, most muscle actin staining. However, in the tumor, you can see in some situations, actually, the tumor green tube is not really supported by parasites. So with all that, if some of us are old enough to remember Judah Folkman, that he proposed to inhibit tumor angiogenesis. That's why you can choke the tumor out of blood vessels so tumor cannot grow. The concept is correct in terms of tumor growth is slower, but increased amount of hypoxia actually, at least in the mouse model, showing that actually cancer cell becomes more motile and metastasis is increased. So since then, I think evolved the concept into tumor vessel normalization. So we do not want to completely block the whole blood vessel inside the tumor. What we wanted to do is to convert the chaotic situation to more normalize the vessel. The advantage, obviously, is if you're treating the patient with drug, the drug delivery will be more efficient, but also normalize the vessel, decrease hypoxia, which inhibit cancer metastasis. More recently, actually, quite a few papers came out showing that normalize the vessel actually is enhancing tumor infiltrating lymphocytes. So there is many ways to normalize tumor blood vessels. Large amount of study has been focusing on the cell surface receptors and growth factors, especially VEGF. The approach we're taking, we're looking downstream of these surface receptors and try to target signaling nodes. In this instance, we are focusing on mTOR. So mTOR is a kinase which can be associated with various different factors to form two large complex, complex 1 and complex 2. So I'm not going to get into the details of difference and similarity of those two complexes for this talk. The only thing that's important to remember is that they have unique components. For mTOR complex 1, we have Vascular Raptor, which is the unique components of mTOR complex 1. So Xiang has made cross of Raptor Flox allele with CDH5, which is the gene encoding for V cadherin, driven CRE-ER. So if we give mice tamoxifen, we'll have inducible endothelium-specific Raptor knockout mice. So when we first studied these mice, we took the endothelial cell out, wild type and knockout. We compare their in vitro proliferation, migration, tube formation, etc. We were a little bit disappointed, which showing that knockout of Raptor or mTOR complex 1 in the vascular endothelial cells, and in vitro, it actually doesn't do much. However, when we put tumor into either wild type or knockout host, as you can see, loss of Raptor is significantly decreased the tumor growth as well as lung metastasis. So this panel A is an LLC tumor allograft model. We also cross these mice to MMTV polyomyelin T transgenic model. This is breast cancer model. As you can see, it basically recapitulates whatever the phenotype we see in the allograft model. So how the tumor blood vessel look like? So it turns out that if you knock out the Raptor in endothelial cell, it increases the integrity and perfusion of tumor vessels, which shown in the panel A is a double staining of smooth muscle actin and CD31. As you can see in the knockout host, that those tumors actually, the blood vessels have more lumen, and the lumen are supported by parasite, which is stained green for smooth muscle actin. And if you inject the lectin intravenously, and you can see the knockout tumor actually is perfused much better. Those are all signs of vessel normalization. Of course, another hallmark of normalization is decreased hypoxia. So there's many ways to measure tumor hypoxia. The one that we choose is to use a reporter construct. So this is mTORCI expression driven by hypoxia responsive elements. So if there is a hypoxia in the tumor, if transcription factor is stabilized, we bind and drive the transcription of mTORCI. As you can see in the lower panel, that those are the immunofluorescence of CD31, and you can see in the white type we have a lot of TORCI expression. However, in the knockout tumor, there is a decreased level of TORCI expression, suggesting that there is a reduced hypoxia level. So far, I've been telling you that the loss of Raptor in vascular endothelial cells will lead to vessel normalization and reduce tumor growth and decrease the metastasis. So when we first got this data, it was somewhat counterintuitive, because I naively thought that you have better vessel perfusion, you have more nutrients, you have more oxygen, it should actually increase tumor growth rather than decrease. So this must be something to do with the immune system, and perhaps that the lymphocytes will come in and will attack the tumor cell. That's why the tumor doesn't grow as much. So we decided to look at the immune system. On the left side is CD45 immunofluorescence, and basically CD45 cells are the pan-immune cells, and it's quite kind of striking. You see there's a lot of green cells in the knockout compared to the white type, and try to be more quantitative. So we did flow cytometry, which is showing on the right panel. We see that the CD45 positive cells are increased, the percentage of CD3 T cells increase, CD4 and CD8 also moderately increased, but more importantly is to measure the effector function of CD8 T cell. In this instance, we use the interferon gamma, which is the pro-inflammatory cytokine, as a marker. It's also increased. So then the question is whether indeed all the phenotype we see is dependent on the lymphocyte. So we decided to deplete lymphocytes using neutralizing antibodies. And this is, we actually did 3 different ways. We started with anti-CD8 alone. We did not see any changes. Then we moved to anti-CD4 alone. We still did not see any changes. However, when we combined anti-CD8 and anti-CD4, the combination actually partially restored the tumor growth in knockout mice, and which is showing in the tumor volume panel in the lower left. So basically, the blue line is the wild type, and the red line is the knockout tumor. And interestingly, if you deplete lymphocytes in the wild type, it does not appear to change very much. However, in the knockout, if you deplete lymphocytes, the tumor grow back. See in the pink line. I think the reason probably is in the wild type you do not have very much lymphocyte infiltration, so if you deplete them, doesn't really make huge amount of difference. However, in the knockout, you have a lot of cells which inside the tumor, and then when you deplete it, a tumor growing back. So the next question is we want to find out how endothelial cell mTOR complex 1 affect tumor infiltrating lymphocytes. So the original idea is try to isolate different cell populations from the tumor and do RNA-seq and try to find the mechanism. I want to say we only partially be successful isolating these cells. The reason was that the tumor cells are very easy to get large, pure population, have high quality RNA. We are still working on the CD31 endothelial cells because the amount of RNA we get is not really sufficient for RNA-seq. But anyhow, so we went ahead, did tumor cell RNA-seq. As you can see, actually not a huge amount of genes are changed. But among the 39 genes got enriched in the knockout mice, a lot of them are involved in the cytokines, showing here CXCL10, GM-CSF, TNF-alpha, increased in the knockout. We decided to focusing on GM-CSF. The reason for that is because it also came out cytokine screen, which is showing in the next slide. So this is the Luminex multiplex assay on tumor lysate. And we have 6 pair of knockout and wild type tumors. As you can see, GM-CSF are increasing all 6 pair, which is further confirmed by qRT-PCR as well as ELISA. So when we're looking into the literature, GM-CSF sort of play a dichotomous role on the immune system. On one hand, there is a large body of literature showing that GM-CSF can activate dendritic cells, enhancing their presentation, antigen presentation to T cells and enhance T cell immunity against the tumor cells. However, there are also many articles you can see in the literature that GM-CSF also recruit and enhance the activity of different myeloid populations, for example, MDSC, which actually can inhibit T cell response. But there are a lot of clinical trials, if you get online, using GM-CSF. It's called GVAC. Those are the 2 examples showing at the bottom panel. The GM-CSF and plus ipilimumab, both in pancreatic cancer as well as in prostate cancer. As you can see that the addition of GVAC then actually significantly increased the patient survival, showing here. So we decided, perhaps in our system, is the GM-CSF is actually stimulating the body's immune system. So the first step we decided to look at is to look at the dendritic cells. So it turns out there is many different types of dendritic cells. If we look at pan dendritic cell with CD45 positive MHC class 2 positive and CD11C positive cell, we did not see any difference. And at the time when we're doing these studies, there are, I think, 3 or 4 papers came out and showing the CD103 positive dendritic cell, which have the circle on the left here, are the one that resides in the tumors and can prime in the T cell in the lymph nodes. In addition, a CD103 dendritic cell also secretes the XCL10, which attracts the lymphocytes into the tumor parenchyma. So we decided to look at the CD103 positive dendritic cells. So indeed, it turns out, as you can see, both in the tumor, which on the right panel G, as well as the draining lymph node in the panel F, CD103 positive dendritic cells, the percentage of those are significantly increased in the knockout mice. So now the question is whether actually due to the GM-CSF. So then we used the neutralizing antibody to remove GM-CSF. As you can see, when we did that, the tumor grew back, partially rescued the phenotype. And also on the right, as you can see, that once we remove the GM-CSF, the CD103 positive dendritic cell population goes back down. So now we updated our model. We think that loss of mTOR complex 1 in the vascular endothelial cell somehow sends a signal. We're still working on it, how that signal goes to the T cells, or not T cells, the tumor cells, to allow tumor cells to secrete more GM-CSF, which attract more CD103 positive dendritic cell, and enhance T cell effector function to attack tumor cells. So we were pretty excited about the translation potential of this discovery. So I thought, because that mTOR complex 1 specific inhibitor, you have FDA approved drugs, for example, rapamycin or Raptor, like RAD001 is already in the clinic. And essentially you can use that to enhance lymphocyte infiltration in the tumor. So I was excitedly telling my son, who's an MD PhD student, I said, hey, we can do this for a clinical trial. And he said, Mom, this is not true. Because if you recall, rapamycin has been used for transplantation rejection. So they will actually have an immune suppression system rather than boost the immune system. So we actually did the literature search, and turns out that he was only partially right. So this is one of the articles. I guess there are several, three or four articles came out during that period of time. This one is in the Science Translational Medicine. So basically the key is at the dose. So if you have the dose that used in oncology or transplantation, basically have immune suppressive effect. However, if you lower the dose, in this article, I think they lowered the dose to 100-fold of RAD001, and they did phase 2 clinical trial in elderly patients. I think they used the vaccination as a measure of immune function, and they found that RAD001 actually enhanced immune function and reduced infection in the elderly population. So we got very excited. We decided to see if we use like really low dose, whether we could selectively inhibit only tumor blood vessels, but without affect immune system. So the answer is yes, and this is the experiment we did. So if you look at the panel A, this is schematics about how we did the experiment. So basically we have LC expressing OVA as an artificial tumor antigen. And at the day 6, we treat these mice with very low dose, 0.01 mg per kg daily injection for 8 days. And on day 14, we adoptively transfer CD8 and CD4 T cells, which are isolated from OT1 or OT2 mice. So these transgenic mice actually express a specific T cell receptor, which can recognize the overpeptide presented on the MHC molecules from the tumor cell. So this response is antigen specific against the tumor response. So in the panel B, showing that if you treat these mice with low dose of RAD001 and tumor growth is much slower if you look at the day 18. And when we isolate various cell population from the tumor, we use the phospho-S6, which is downstream target of mTOR complex 1 signaling as a marker for mTOR signaling. And as you can see in the panel C, basically it's CD1 positive vascular endothelial cell. If you treat with low dose RAD001, phospho-S6 is significantly decreased. However, this is not true in immune cells or tumor cells in the other population. Showing here CD31 minus, CD45 minus cells. Those are mainly tumor cells and fibroblasts. So there is no decrease. And CD45 positive cells are immune population. There is no decrease. So this shows that if you treat these mice with low dose RAD001, they are only selectively inhibits mTOR complex 1 signaling in the tumor endothelium. So how about vessel normalization and T cell function? So we did very similar study using immunofluorescence staining of CD31 and smooth muscle actin. As you can see, very much like in the knockout tumor RAD001 treatment, which create the lumen, suggesting better perfusion, and the vessel is more better supported by smooth muscle actin expressing parasites, indication of normalization of the tumor blood vessels. And if we look at the lower panel, is the flow cytometry analysis, looking at first the CD45 infiltration, and you can see in the low dose treatment, CD45 immune cells are significantly increased in the knockout, not knockout, in the RAD001 treated, as well as CD8 T cell. If you get down the CD8 T cells, is also increased in the RAD001 treatment group. Those donor cell also contains a reporter, YFP, driven by interferon gamma promoting enhancer. So with high YFP, which is indication of better effector function. And again, you see that with the low dose treatment, also significantly increased CD8 positive T cells expressing interferon gamma. So I think this is have real potential in terms of how to prepare the tumor for infiltration of lymphocytes. Perhaps it could be used as a combination with other treatments, for example, adoptive T cell therapy or a checkpoint inhibitor. So to summarize this part of talk, we show that loss of mTOR complex 1 in the vascular endothelial cells normalize tumor blood vessels, inhibit tumor growth and metastasis. We think mechanism, at least in part, is due to elevate the CD8 positive T cell effector functions, and is through partially through JChen mediated activation of CD103 positive dendritic cells. And as I just mentioned, this also have translational potential because the RAD001 is FDA approved the drug, and low dose can selectively target the endothelial cell without affecting tumor cell and immune cells, and perhaps you can use it in combination with other immune therapies.
Can we ask a quick question before you go on to the next?
Sure.
Yeah. So that was quite impressive. I'm just wondering what kind of tumor cell line are you using for your experiment? So are these from carcinoma cell lines or sarcoma or lymphoid?
Okay, yeah. So we use the two different lines. We use the ALC, Lewis lung carcinoma line. We also use the polyoma middle T, which is the breast cancer line.
Okay.
For those studies. So two different lines. We have crossed the Raptor allele into the MMTV polyoma middle T, but we did not do the immune analysis in that line. The reason for that is they just come, tumor comes at different times, so it's kind of hard to get enough mice to analyze it. So all the immune studies are in the sort of transplantation model.
Mm-hmm. Yeah.
I have a question on this. I'm sorry, Deli, go ahead.
Yeah, sorry, Eddie. My second follow-up question is in the role of VEGF in all this. So, some of the cell lines you talk about, these are tumors that are known to respond partly to VEGF inhibitor. So now from your data, by restoring the vascular structure of these tumors, you actually improve the outcomes. So what will be the role of VEGF in the whole process that you just described?
Yeah, so that was a really good suggestion that we have not directly tested in terms of VEGF, but I think that actually mTOR complex 1 is directly downstream of VEGF. So I think perhaps you use neutralizing antibody against the VEGF, you may also see similar effect. I think that was very good comment. However, I want to point out mTOR complex 1 probably is also downstream of other receptors, for example, FGF receptor. So I think if I remember correctly, that years ago that Dr. Hanahan's group have shown that if you inhibit the VEGF for angiogenesis, there is a compensatory mechanism for FGF and other growth factors. So the hope is we have mTOR complex 1 downstream of VEGF, but also downstream of other receptor, maybe not, although we did not directly test it, maybe we have less kind of compensatory mechanism that tumor we use. This is just, speculation.
Dr. Chen, one quick question, Leo Fert here. In the early experiments you showed with the Raptor knockout and the decrease in tumor growth and metastasis, if you let those animals live longer, does that change? Is it just slowing down the process, or is it in fact, if you run it out, the tumors always stay at a lower level unless metastasis?
Unfortunately, it's a very good question. Unfortunately, we have not let these animals, go beyond whatever shown the day.
Fine, thank you.
So I may have some compensatory mechanism come back, but we don't know.
Thank you.
Okay. So the second story is based actually on the concept of tumor cell and lymphocytes utilize the same nutrients. This is actually fairly well studied in terms of glucose metabolism. I think there's quite a few papers several years ago showing that the tumor cell can outcompete T cells for utilizing the glucose. So tumor cell enhance the glycolysis, deprive of T cells glucose, and inhibiting T cell effector function. So we saw kind of analogous to glucose metabolism. Perhaps there is also competition for glutamine between the tumor cells and T cells. So just kind of a brief reminder of glutamine on the left panel. So glutamine is taken into the cell by transporter, showing here the SLC1A5, which encodes for ASCT2. Glutamine can be utilized for nucleotide as well as amino acid synthesis, but also glycosylation. And glutamine in the cells can be further converted into glutamate via glutaminase. And glutamate is a precursor for glutathione, which is a major cellular antioxidant. So glutamate can be further converted into alpha-ketoglutarate, which will join the TCA cycle, either produce the ATP or funnel to lipid biosynthesis. But you guys probably also all know that the alpha-ketoglutarate is a substrate for dioxygenase, which modifies DNA and histones, kind of involved into epigenetic regulation. So the first thing we do is on the right, and we show that we want to see whether actually glutamine level can make an impact on T cell activity. So in this experiment, basically we just isolate CD8 positive T cell. We will activate with CD3 and CD28, and then we throw them in the different concentration of glutamine. I wanted to say glutamine actually was pretty abundant amino acid, like in the bloodstream, so close to 2 millimolar concentration. However, in the tissue, there is a couple of papers in the tissue could be like tenfold less, more like in the 0.1 or 0.2 millimolar. So as you can see, after 4 days, we measure the interferon gamma production by ELISA. You can see that those cells growing at 2 millimolar high glutamine concentration, they produce a lot more interferon gamma, suggesting that the CD8 T cells are relying on glutamine metabolism for their best effector function. So to test whether actually there is competition, and we used triple negative breast cancer because in the literature that triple negative breast cancer was shown to be addicted to the glutamine metabolism. So to do this, we knock out glutaminase in E0771 line, which is a triple negative line shown here. And then we implant these cells into the mice. And you can see that knockout of glutaminase in the tumor cells significantly decrease the tumor growth as well as lung metastasis, is shown here. But the interesting part is this is only happening in the white type mice, which is immunocompetent. When Diana actually did a side-by-side comparison in the RAG1 deficient mice, which is lacking of mature B and T cells, and you can see the tumor growth actually is kind of similar, which is an indication that the lymphocytes are required for tumor growth defect in glutaminase knockout allograft. So we then went on trying to test with this is also true in a transgenic model. So Diana have crossed GLS flox allele with C3 tag, which is a mouse model for triple negative breast cancer. She also crossed in MMTV-CRE, which knock out glutaminase in the mammary epithelial cells. And in the bottom panel shows that the knockout have significantly increased the tumor latency, have better survival, and decreased the tumor volume. So now the question is what happens in the immune cells? The prediction is the immune cells will actually function better. So it turns out indeed that memory epithelial cell specific knockout of glutaminase actually improves CD8 T cell effect function. So here on the left most panels showing that the CD4 and CD8 percentage of population increased in the knockout. And as you all know, that CD8 exerts their cytolytic functions through release of serine protease granzyme B. Showing here the CD8 expressing granzyme B, the populations are increased, as well as expressing CD107 is a degranulation marker, or interferon gamma, which is pro-inflammatory markers, are all increased. It's showing here. And so our results suggested that if you knock out tumor cell glutamine metabolism, you have better T cell function. And this model also predicts if you knock out glutamine metabolism in cancer cell, you will have increased the glutamine, perhaps in the tumor interstitial fluid. So to test that, we used a very simplistic assay. Basically, we got the tumor, we put them in an envelope with a filter, then you spin them really slowly, and we get tumor interstitial fluid. And then we assay the glutamine level inside the fluid. So as I mentioned earlier, indeed, in the tumor, I think the tumor interstitial glutamine level is quite low. Showing here it's about 100 micromolar. However, if you delete JLS in the tumor cells, the interstitial fluid glutamine level are significantly increased, and it's more approaching the serum level of 1 millimolar. So how do we targeting the glutamine metabolism in cancer? As you probably all know, that the various different inhibitors has been developed for inhibiting the glutaminase. The CB compound actually is a clinical compound, have been through various different clinical trials. I think the results from the clinical trial is this CB compound is very safe, but as a single agent is not terribly efficacious. So the company is trying to see if doing any combination therapy trials. During the meantime, Charles Manning, at that time he was still at Vanderbilt, he developed a compound called V9302. Is relatively selective for glutamine transporter SLC1A5. He actually, in their Nature Medicine paper, he actually directly compared side by side with V compound and CV compound, and showing V compound is much more efficacious. So we decided to try his compound. And we took his compound, we let tumor grow for 12 days. Then we inject for 5 days with his compound. And indeed, it's quite potent. As you can see, that the tumor growth almost, basically stops there. So this is kind of encouraging. It's very efficient compound, but also kind of at time it was a worry to us, because we think, both tumor cells and T cells that eat glutamine, and now you have this transporter inhibitor there, probably, certainly inhibits tumor cell, but also inhibits T cells. So we want to look at what's happening with T cells. So to our surprise, that T cell actually not only is not inhibited, their function is going up. On the left panel, we're showing that actually the number is slightly decreased, but effector function, as measured by granzyme B expression or CD107A expression or interferon gamma expression, is all increased in the presence of the V compound. So we decided to actually look at CD8 cytotoxicity, and the most lower panel is the co-culture of EO771 expressing OVA as a tumor antigen, and co-culture with the CD8 T cells that are isolated from OT1 transgenic mice. And these CD8 T cells express a specific transgenic T cell receptor, which will recognize the overpeptide on the EO771 and presented by MHC to exert the killing. So anyhow, to make a long story short, we can see actually in the presence of V9302 compound and actually the tumor cell killing actually is quite a bit of increase, suggesting a better T cell effector function and cytotoxicity in presence of V9002 compound. So what is the mechanism and why you have this transporter inhibitor of glutamine and T cell actually can function better? So first thing we did is we look at the glutamine uptake because this is transporter inhibitor. So we use the two systems. We use the murine EO771 with CD8 T cells isolated from mouse. We also use the human system with HCC 1806 with CD8 T cell isolated from healthy donor of human. So in both instances, interestingly, this V compound is really inhibited uptake of glutamine in the tumor cell, but if anything, it's enhanced the glutamine uptake in the T cells. So this is kind of interesting to us. So we decided to look at all transporters. So as you can see, there is a number of transporters can transport the glutamine, which is showing on the left in a review. So Diana actually tested each of them to make a long story short, and she discovered that the V compound induced expression of ATB, which is encoded by SLC6814 in the CD8 T cells, but not in the tumor cells, showing here the EO771 and 41. On the top panel is the qRT-PCR, showing the elevated expression of SLC6814, and the bottom panel is the Western blot analysis and showing the ATB protein level is increased in the presence of V9302 compound. So now the question is, this transporter is increased, whether actually it is the one that is responsible for increased uptake in activated T cells. So Diana did two things. The first one, she's knocked down ATB in the CD8 T cells, showing on the left, with two different sequences. As you can see, once you knock down, and the elevated level of uptake in the presence of V compound is back down to the control level, showing here. And conversely, when she overexpressed SLC6814, showing here, the uptake actually in the CD8 T cells actually is increased. And she also did cytotoxicity assay, and very similar to the one I just described, and showing that the overexpression of ATB in the CD8 T cells actually increase the cytotoxicity. So we think this is the responsible transporter for glutamine uptake in the CD8 T cells. So how influx of glutamine affect the T cell effector function? So Diana actually did various different experiments. Some of them are not working, but we're showing the one that is working here, which is summarized on the left panel, showing that in the presence of V compound, you have increased the level of the transporter ATB, and once inside the cell, it can be converted into glutamine, can be converted into glutamate. And glutamate can exchange for cysteine through this antiporter, which is also increased in presence of the V compound. And then cysteine and glutamate are precursor for the antioxidant glutathione, which is synthesized by this enzyme, glutamate cysteine ligase, showing here, the level of which is also increased, which is showing on the right panel. The glutathione level is increased, and intracellular cysteine level is increased, the antibody expressions increase, this enzyme glutamate cysteine ligase levels increase, and last but not least, the loss level actually is decreased. So we think that V compound is improved. V compounds have this influx of glutamine to improve the redox status of CD8 T cells. So to summarize this part, we show that loss of glutaminase in the tumor cells or memory tissues delayed tumor growth and progression by enhancing CD8 T cell effector function. We think selective targeting glutamine metabolism, either by knockout in the tumor cells or by V9302 compounds in tumor cells, increase glutamine availability within the tumor interstitial. So allowing the T cell actually uptake more glutamine in the presence of the V compound, which is the mechanism of which actually is compensatory induction of ATP transporter. And I just during the last slide that the influx in the presence of V compound supports de novo glutathione synthesis and improves redox balance in T cells. So, as you're all aware, there's many different ways to improve anti-tumor immunity. You probably heard about, through combination of targeted therapy or chemotherapy to release the tumor antigens, or to enhance interferon gamma signaling pathways, or enhancing MHC production expression in the tumor cell, etc. So today is the story. I'm just showing you two additional ways. One, you can improve tumor micronormalization, as well as trying to selectively inhibit tumor cell metabolism. I think either glutamine metabolism or glucose metabolism will change the immune microenvironment, allowing T cells to function better. So I should stop here. I already mentioned that people have done the work. Diana is on the glutamine studies, and Sean was on the mTOR complex 1 in the endothelial cell studies. And we have this wonderful group of collaborators, Mark Boothby, who has a lab just upstairs from my lab, who is an immunologist, and we did all those immune analysis in collaboration with their lab. The CDH5 CRE-ER, the transgenic mice was originally made in Ralph Adams' lab in Germany, but we got these mice from Ho Sun, who is right now at the Harvard Medical School. Charles Manning supplied us with V compound. Unfortunately, he actually got a really good position, so he moved to MD Anderson right now, although we are still collaborating. Last but not least, these works are funded by NHR1 and VA, as well as Common Foundation. So I stop here. We love to answer questions. Hopefully I did not go over.
Thank you very much. It's a very stimulating talk. So I'd like to invite questions from the audience. Please go ahead and identify yourself and ask question. Maybe I can start off. I was curious when F proteins bind to the Fn receptors. How does the activation occur? Is that autophosphorylation, or there some other substrates that are phosphorylated?
Yeah. Okay, so when it's bind, it's not binding a single molecule. They usually have a cluster. I think that several years ago, they have very nice structural analysis and showing that once the receptor clusters in tetramer or more than tetramer, actually, it can transphosphorate each other. And activating SARC, activating exchange factors like CD, and then activate CDC42 and ROA, etc. So signaling pathways, I think, is reasonably well characterized. In terms of a story connected with the glutamine, and we show that the FA2, or I want to say, in the tumor cells, and actually is overexpression of receptors is phosphorylate itself. So it's a ligand independent signaling in the tumor cells, and we show that you can actually enhance the expression of this glutaminase as well as the transporters is by activation of the signaling pathway YAP and TAZ through the ROA activation. So there is many different pathways, and once you have the ligand there, it actually have an inhibitory role. So F receptor is quite actually complex in terms of ligand dependent and independent signaling. So once you have ligand, it's usually to activate a pathway which will degrade the receptor. So after you treat the ligand stage, the receptor will be gone. Once running with it, can't see it. So ligand dependent signaling actually play a role in inhibitory role. However, ligand independent, which in most the cells, tumor cells, employ is having a tumor promotion. So it's this dichotomy effect of EPH receptor.
Thank you. Other questions?
Yeah, I can. So thank you again, Dr. Chen. I think that was quite stimulating. So as you know, one of the problems with modifying T cells, effector T cells against tumor is that it's non-selective. So it kills the tumor, but then there's always the side effect of, causing autoimmunity and damage to non-tumor cells. So with the glutamine synthetase and the CD8 cells, so do you foresee a way where this can be harnessed in a very selective way, or is it going to be something that you expect more of a systemic side effect?
Yeah, this is a really good point, because the side effect is a really problem in terms of immunotherapy, right? Yeah. So I think we want to have selectively targeting the tumor without affecting the normal tissue, I guess. Is that your question?
Yes, that's really what.
Yeah, so there are actually quite a, right now, as a lot of people try to study that in terms to see if any shared epitope or whether T cell clone, whichever T cell clone will attack the normal tissue. Actually, I was sitting on the spore panel, and actually a lot of NCI spore programmers exactly study this. I should say, right, currently I don't have a perfect solution to how do you avoid the side effects. But a lot of people studying.
Yeah, I mean, I think this gives you one of the unique opportunities. So if you look at all the other efforts, this has been at the level of the CD4, at the antigen presenting level with the PD1 and CTL4. So now you are looking at targeting the effector cells, which is more downstream influence. So maybe there will be a way to, first expand the T8 cells that you have in mind, and then be able to modify there. So, I mean, I know it's early days, but it's quite, impressive.
Yeah, it's almost you want to have something. I think adoptive T cell therapy, maybe eventually you can do that. If somehow you could, I don't know, somehow you could have a switch on and off system. You switch on and you attack tumor cells, and somehow you can engineer in the T cell, you can switch it off. I think that kind of is possible. I can see it can be like engineered in the T cell therapy. In terms of immuno checkpoint inhibitor, presumably, if you stop treatment, I think in the clinical sense, even if you stop treatment, it's still actually those T cells sometimes still active. You know, how do you stop those ones?
Thank you.
Well, this is this is a very interesting problem. I think a lot of people working on this, and hopefully we will solve it to have selectively target only tumor instead of normal tissue.
Thank you.
Any other questions? But, if not, I think it's 9 o'clock. We like to thank Dr. Chen again for taking the time to present this Grand Rounds talk. I really appreciate it. Thank you.